Okay, so the next section will be on fluid movement across compartments. So please make sure that you go back and review when we talked about like capillary dynamics and capillary dynamics at the glomerulus. And just make sure that you review the general terms that you should know. So like hydrostatic pressure, which I think this is already in your note. So I'll just say HP because yes, it is already in your notes. Hydrostatic pressure is a pushing force usually caused by blood pressure. And then osmotic pressure is the pulling force or the attraction of water towards areas that are more concentrated. And so solutes move according to their concentration gradient, and their charge and their transport proteins. And so solutes move from high concentration to low concentration to make sure that the solutes are in equilibrium and even on either side of a membrane, while water tends to move from lower concentrations to higher concentrations or higher concentrated areas to make sure that the concentrations are equal on either side. So water moves to a higher concentration to dilute that solution so that it can be equal to the lesser concentrated or the less osmolality side. And so here are the three fluid compartments. We have blood plasma in red here, interstitial fluid in blue here in the middle, and then our intracellular fluid inside the cells here. And so there is a little bit in your lecture notes about exchanges between the blood plasma and the interstitial fluid. So you should know what the barrier is between the blood plasma and the interstitial fluid. And we talked about this before. It is the capillary. So the capillary is going to be, or you can say the capillary wall. Capillary wall is going to be the dividing line between our blood plasma and our interstitial fluid, while the dividing line or barrier between our interstitial fluid and our, inside of our cells is the plasma membrane. Okay, so those are our barriers, and that's how fluid is going to move. So when we have new oxygen brought in from our lungs, it goes into the blood plasma, across the respiratory membrane, into interstitial fluid, and then into cells. And CO2 going out will go from cells into the interstitial fluid, eventually to the blood plasma, and then eventually out to our lungs and out the body. Nutrients, again, as we're eating new food, introducing new foods to our body, those get broken down into their monomers, and then they move into the blood, which then moves through the interstitial fluid into our cells, so our cells can use those monomers and the ions and such that get absorbed in our cells for cellular work. And so just make sure you know how the movement occurs. So from blood plasma to interstitial fluid, it has to move across a capillary wall, whether it's a continuous capillary or fenestrated capillary or sinusoid capillary. And then things or water that moves from interstitial fluid into the cells or vice versa is going to go across that plasma membrane. So yeah, I think that's all I wanted to say there. So then the next slide is all about osmosis, which just should be a review. And so I just gave the answer to that one. But when there's a lower concentration of solute outside the cell compared to inside the cell, we call that cell to be in a hypotonic solution. So when a cell is in a hypotonic solution, that means the concentration outside the cell is less than the concentration inside the cell. When a cell is in solution, and the concentration outside the cell is equal to that inside the cell, we call it isotonic. That cell is in an isotonic solution. And then when a cell is in solution and the concentration is higher on the outside of the cell compared to the inside of the cell, we say that that cell is in a hypertonic solution. And so now we're going to talk about the movement of water. And so in a hypotonic solution where the concentration is less on the outside and it's more concentrated on the inside, water tends to move towards the higher concentration. So let's say inside the cell, and I'll make a little drawing over here. let's say inside the cell, it's at, I don't know, 4%. I will say sodium chloride. These are just random numbers I'm giving and I'm drawing with my mouse, so please be patient with me. So I'll say inside the cell, it's 4% sodium chloride. Outside the cell, it's only 2% sodium chloride. And so the water will tend to move inside the cell, which is going to bring this concentration down and then this concentration up. And I really wish I had a better tool to draw on this, but I'm on my laptop. And so again, if the concentration outside the cell is less than the concentration inside the cell, water will move towards the greater concentration. And so in that case, that would cause the cell to swell, okay, as it fills up with water. And we talked a little bit about water toxicity. If it's too much of a difference, if that hypotonicity outside the cell is much different than the concentration on the inside of the cell, then that red blood cell, if we're talking about a red blood cell, can only withstand so much pressure. Our cells are human cells, and so they don't have a cell wall like a plant would that can withstand more pressure. No, we just have our plasma membrane, and so it can only withstand so much pressure. And if we move too much water into that cell will burst, which is why water toxicity can eventually lead to death if we have all of our red blood cells lysing. In an isotonic solution where concentrations are equal on both sides of the membrane, is the water going to move into the cell, out of the cell, both directions? Is it not going to move at all? And so the answer to that is that it actually will move in both directions. Water will never, ever, ever stop moving. Okay, water is in constant movement. So we never want to have things just stop moving altogether. So what's going to happen in an isotonic solution is that the same amount of water that's going into the cell is going to be leaving the cell at the same rate. And so we keep those equal concentrations, but we also are keeping water moving. So we're moving water into the cell at the same rate that we're moving the water outside the cell. And this should have no effect on the shape of that cell. Water is moving equally in both directions. It should stay its normal shape and size, which in the case of a red blood cell, it's going to stay its nice biconcave shape, and you won't get any swelling or shrinking or anything like that. It'll stay its normal size. In a hypertonic solution, where the concentration is greater on the outside of the cell than it is on the inside of the cell, so we're going to have a opposite kind of situation happening here. So let's say inside the cell now it's 2%. Oh my God, this is so bad. 2%. And then outside the cell, it's greater at 4%. Then that water is now going to want to move out of the cell to dilute that higher concentration, bringing this concentration down. And by removing water inside the cell, it's going to make that concentration go up until hopefully they reach an equilibrium. But this cell is still losing water. And so if it loses water, like it would with a hypertonic solution, that cell is going to shrivel, or it's also called crenation. It's also going to crenate. Okay. And either way, if we are either swelling the cell to a point where it lyses, or we're shriveling the cell, crenating it, or shrinking, all those words all mean the same thing. But if we are shrinking the cell or swelling the cell, both of those can have harmful effects. Okay, we want to be in this isotonic equal movement of water kind of situation. So I have these little images here. So remember, hypo makes a hippo. So if you're in a hypotonic solution, that's when water moves into the cell, swelling the cell like a hippo, okay? Or in a hypertonic solution, it's going to be the opposite, and you're going to have shrinking of that cell. Okay. So make sure you review your osmosis. This is just the same thing, but in a different diagram or visual here. So isotonic solution, water is moving in either direction. And so a biconcave shaped red blood cell would stay a biconcave shaped red blood cell. In a hypertonic solution where there's a greater concentration on the outside of the cell and water moves outside of the cell to dilute that higher concentration solution, that cell is going to crenate or shrink or shrivel. You can use any one of those words. And the cell is going to be made smaller, which is going to make it harder for it to carry out its functions. In a hypotonic solution where the concentration is less on the outside, greater on the inside, water is now going to want to move towards a higher concentration into the cell, which is going to swell that cell, which is also going to mess with function. And if it swells past its capacity for pressure, then that cell is going to lyse. All right, and this is where I'll stop here, and we'll do the next video on intake and outtake of water and its regulation.